Okay, let's get started. Today we have two lectures. The first one will be about principal component analysis, and in the second one we'll do an overview of everything we've covered so far and see how it relates to each other. And that's also an opportunity for you to ask questions so we can answer them too, and so everyone can hear them. Okay, the topic of today is principal component analysis, and after this lecture we have about 2 hours to do a practical on this. The idea is just to fit a couple of PCAs and see what it looks like and try and plot it and see if you can, extract any meaningful information from it. The exercises are over here, so the website we've been using, and then 11.5.1, and it works again by downloading something and then opening it in RStudio. There's no instructions on how to do PCA, but they are written over here in the book. You can just download it, and then paragraph 4.4, there's a complete example how to do it in R. So you can just copy that and then perform it on the data given in the question. I will show the example with the from this book at the end of the lecture. Okay, I have to face this way because I also had problems with the Wi-Fi today. So if I miss your question, just make some noise or something. Okay, today's topic is principal component analysis. This is a very useful tool. It's a kind of exploratory data analysis. Let me confirm that people can hear me. Okay, no comments yet. I'll take that to mean that people can actually hear me. So PCA is not like the other things we've been doing. It isn't really a model, and you can't do significance tests with it, and you can't use it for prediction. The only thing it's for is like summarizing your data. And so far we've been working with really small data sets of usually like 2 to maybe 5, maybe 10 variables, but that's it. But in practice you'll often have to deal with much larger data sets than that. And then it becomes kind of hard to see what's going on. Could there be interactions? Could there be clusters in my data? So that's what PCA is for. It's to summarize high-dimensional data. Okay, I've divided into 4 different parts. First, I'll talk about why you would use PCA, because I want to convince you that it is useful and it's maybe something you should do at the beginning of any analysis that involves a large data set. Then I'll explain the terminology. So we're all on the same page. So what are principal components? What are the scores? What are the loadings? And then I'll go over an example. And because we are talking about high-dimensional data, it's a bit difficult to follow along if I start with a real data set, which is large. So I'll start with a 2-dimensional data set with only 2 variables. Now, nobody does PCA on 2 dimensions. It makes no sense, but it's just so you can see what's happening. And then from 2D we go to 3D, and then finally from 3D we'll go to 4D, because that's something I can't plot, and then you can see, hey, PCA is actually useful. And finally I'll talk about the choices you can make in doing PCA and why you would choose which. Okay, so why would you use PCA? For the purpose of this course, the only thing you have to know is that you can visualize high-dimensional data with it. But you can do many other things with it. You can use it for dimension reduction. So maybe you have 20 variables and you're like, well, can I maybe reduce this to 10 or to 5? Can I use less variables for my problem? That can be done with PCA. And also if your data are very highly correlated or collinear, so that means not that the outcome is correlated to the explanatory variable, but the explanatory variables themselves are correlated to each other. That's actually a big problem, because that's one of the things that causes collinearity. And using PCA, you can turn it into a regression of uncorrelated variables, which is called principal component regression. But you don't have to remember that. The only thing we're going to do now is visualize high-dimensional data, and that's also the only thing we're going to do in the practical. Okay, so why would you use PCA? Let's say that you're at the zoo, and yeah, there's all kinds of animals, and they have different colors, they have different heights. They may have 2 legs, they may have 4 legs. There's all these different properties that we could describe of the animals, right? And the question is, how can we take a picture? From which direction should we take a picture such that we have the largest variance in animals in the x direction, and then the second largest variance in the y direction? So how can we get a very diverse picture of animals if we only get to take one picture? What angle should we move around the zoo, maybe from a helicopter or something, that we get this picture with the maximum variance? That's what PCA does. Okay, so how would you apply this to a real problem? Let's say that you have some gene expression data, and it involves 40 genes. Now that's already quite a lot to be producing summary tables and correlation matrices, because you'd be looking at how 40 genes interact with themselves, with 40 genes, but you could imagine the number of interactions becomes insanely large, and 40 isn't even really considered a large data set. If you work in metabolomics, it's quite normal to have in the thousands of metabolites. I did my PhD at a proteomics lab where it's quite normal to have in the hundreds. And then people ask, how do they interact? Well, then you're in the tens of thousands. So yeah, data sets can get really large really quick. And how do you know there's an outlier? How do you know there might be clusters? It's really impossible just by scrolling through a very large Excel file. It's not going to give you any knowledge. So we need some sort of tool to visualize this high-dimensional space, and that's what we're going to do with PCA. Yeah, so there's a point where just looking at the data frame is not only inefficient, but also you cannot remember that many numbers to actually make any connections. So for very large data sets, all the things that we always do at the beginning of an analysis, like called a structure with str, head, that will give you the first couple rows, and summary, that will tell you, this one has this minimum, this maximum, this median, this mean. But yeah, if you have a large enough data set, even those aren't really informative anymore, because there's just too many things to consider at once. So why is it called principal component analysis? Because we are determining what the principal components are. This is a method for finding the largest variance using linear combinations of your input. So the way to put that into perspective with what we've been doing so far is, imagine you have a data set with no outcome, so only explanatory variables, and you want to know if there's a possible linear relationship between those explanatory variables, such that we can simplify the description of our data. Now, what does linear mean? Yeah? Yeah, exactly. So it means that there might be some combination of explanatory variable 1 times this, plus explanatory variable 2 times that, plus explanatory variable number 3 times this, etc. That gives a better summary of the total data set than just using variable number 1 or variable number 2. So that's what linear combinations means. Okay. These linear combinations are called principal components, and a very important property is that they are orthogonal. So that means they carry unique information. So the first principal component will have the largest variance in your data set. The second one is perpendicular to it. So it describes different information in your data. Yeah, because you can be more to the left or more to the right. That says nothing about how high or low you are. So it's important to note that they carry unique information. Okay, the location of your observations, because we can actually plot, your observations in this new coordinate system that we create. That's called the scores. And then finally, the contribution of the original variable. So I said, you do this times the first variable, plus this many times the second variable, plus minus 1 times the third, etc. So they have some contribution to each principal component, and those contributions are called the loadings. Yeah, this will all make a lot more sense when I show what it looks like, but it's just these are the terms about principal component analysis that you should know. Okay, so PCA is used when your data has many variables. So we don't ever use it for two-dimensional data sets, but this is just to see what's happening. So we're going to do PCA on a two-dimensional data set. So what is the setup here? Imagine that we have some plasmid containing a gene for an antibiotic and also GFP, so green fluorescent protein. And we're going to look at the GFP expression, maybe measuring fluorescence or something. And we're going to measure the diameter of bacterial colonies to see, okay, how successful was the transfer of this plasmid, and do bacteria with the plasmid, so the ones with high GFP expression, are they smaller or bigger in diameter than the ones without? Yeah, so we have two variables. We have the colony size, the diameter, and we have the relative GFP expression. So let's see what PCA looks like. Here's our original data set, and apparently the more GFP is being expressed, maybe this costs a lot of energy, causes the colony size to be small, yeah, because here we are on the left of this axis, the colony size, and here we are at high GFP expression, here we are at relatively low GFP expression. I've also colored it, so yeah, the brownish ones are low GFP expression, and the green ones, the brighter they are, the higher GFP expression. This will stay the same throughout the other examples. And the size of the points represents the colony size. Yeah? Of course, this is not necessary to give it colors and to give the symbols different size, because we already plotted both dimensions. But once we get to 3 or 4 dimensions, this will be useful. Okay, what does PCA do? It finds the largest variance among combinations of variables. So that means we cannot just move in the x direction or the y direction, but we can move in any direction that is a linear function of both this and that one. So that means we can find the largest variance, which is like this, right? I hope you'll agree this is the largest variance. And then perpendicular to that, we can make another principal component, which explains the remaining variance, yeah? That's the red line. Now, then I can plot my observations as scores in this new principal component system, and then this blue direction becomes the new x-axis, yeah, which is principal component 1, and this remaining variance in the perpendicular direction becomes the y-axis, which is principal component 2. Now, what we can see here is that indeed it found the largest variance in the first principal component, because this direction already covers 83.7% of the total variance in this data set. And there's only 16.3% remaining in the second direction. Yeah, so there we see what PCA does. Is that clear so far? Okay. Yeah, so it's just a repetition of the question in case you forgot what it's about. But let's say that we're going to measure something else. So not just the colony size, not just the GFP expression, but let's say that these bacteria also produce a halo around them, because they are, producing antibiotics, and maybe nothing can grow within a certain area from the bacterial colony, producing a halo. So now we have 3 different variables. We have the colony size, the diameter, we have the relative GFP expression, and we have the halo size. Let me quickly check if there are any comments so far. I don't think so. Okay. So now we have 3 variables. Yeah, that can still pretty easily be plotted. Just do 2 by 2 of each of your variables, right? So here we have colony size on the x-axis, GFP expression on the y-axis, and we can see, okay, there's a negative correlation between them of about 70%. We can do the same for colony size versus halo size, and we can see, well, okay, these aren't really correlated, and it's very small. It's only 20%, right? And we can express the last combination, which is the halo size, against a GFP expression, which apparently is positively correlated, meaning that the more GFP is being expressed, of course, the more antibiotic it produces apparently, and thus it produces a larger halo apparently. Now, for 3-dimensional data you can of course still do this, but for 5 or for 6 or for 10-dimensional data, the number of combinations of 2 you can make becomes really large, so this isn't a viable option anymore. So let's see what PCA looks on that. Here we have fake 3D perspective. what have I done here? I've plotted on the y-axis the halo size, on the x-axis this is the z-axis, and on the x-axis the colony size, and on the y-axis the relative GFP expression. So now we have 3 dimensions. Coloring scheme is still the same, so the brown ones have lower GFP expression, the green ones have higher GFP expression. The halo is represented by a circle around the observation. So yeah, plot becomes really crowded and becomes a bit subjective how to interpret this. You know, is this one really high in halo size, or is it just far in the back? Yeah, maybe if I use some programs I could turn it around and we could inspect it. That works. But you can see where this is going, right? For 10-dimensional data, there really is no way to summarize it. So let's do PCA. It finds the largest variance among any combination of these 3 dimensions. So it can move in any direction linearly in these 3 dimensions, which is this blue direction. That apparently explains the largest variance if you can only move in one axis. Okay, that one is now over here. That's the x-axis. And apparently that already explains 63.2% of the entire variance in all 3 dimensions. Now, then the second one must be perpendicular to the first one. But since this is 3-dimensional, perpendicular can be anywhere in a circle around this first line, right? I can be perpendicular like this, but also like this and like this, all the way around my first axis. So it finds the largest one possible that is still perpendicular to the blue one. Yeah, that is the red one. Now, and here we have it. Now I have a 2-dimensional plot that explains 62.2% plus 27%. So we have captured about 90% of the data, of the variance in the entire data set using just a 2-dimensional plot. So this would be a nice summary of this data. Okay, so it finds the largest variance in the data, and that we call principal component 1, PC1. And then it finds the largest variance perpendicular to the first direction, and that we call principal component 2, and so on. Now, using only these 2, we've already explained 90% of the variance in our data. And yeah, that means that almost all of the variability, almost everything going on in our data, we can see in this one plot. There's only 10% unaccounted for. Yeah, we can increase the how informative this plot is by turning it into a biplot. So that's the next thing I'm going to show. What is a biplot? Which is how you would usually plot the results of your principal component analysis. Okay, so these are the scores, right? This is actually the same figure that I show over here on the right, just without any coloring. These are the scores. We have this first principal component, which explains 60-something%, and this one, which explains 20-something%. Now, we can also plot the contributions of the original variables in this same figure, like this, right? So actually, if you're on PC1, that one, yeah, if you go up in PC1, then the relative GFP expression also goes up, and the halo size also goes up. If we go down towards the negative part of PC1, then the colony size goes up. Yeah, and we can see if you have a larger colony size, then you have lower GFP expression because they are in opposite directions. And we can see that the halo size is not really directly correlated to either of the 2. Yeah? So now we can superimpose one over the other, and that's what we call a biplot. Yeah, it shows both where your observations are, so you can color them. You can say, okay, if I have male, okay, there are male and female bacteria, but for the sake of the example, let's say this would be a large group of males and females. I can say, okay, color all the males blue, and then I can see, do they cluster somewhere? Are they somewhere different than my females, for example? Now, that is what we can use it for, and now we can do it with even more. Yeah, you don't really need PCA for 3 dimensions, but what if I have 4 dimensions, or 10, or 100, right? I can plot 1-dimensional data, it's just along one axis, 2-dimensional data along 2 axes, and yeah, you can use a fake perspective to get a 3-dimensional plot. But yeah, 4-dimensional, there isn't really a way to plot 4 physical dimensions without involving time or something. So yeah, that's a bit hard to imagine, but it's very easy to do in a biplot. It looks exactly the same. There's just one more line which represents the contribution of this fourth variable to my principal components. Yeah? Now we have the colony size, the halo size, the relative G expression, and the milligrams of carbon that was in their in their medium. Yeah, it's just another direction in my plot, meaning that if I go more in this direction, okay, apparently larger colony sizes also had more carbon in their medium, which makes sense. And that's how you can interpret this. And this plot explains 77 plus 14.2% of the variance. Again, over 90% of the variance in this data set, which is 4-dimensional, represented in this nice 2D figure. So that's what Pca is for. Yeah? It's easily interpretable even though we can't plot 4 dimensions. And it explains a large part of the total variance in your data. So you could draw conclusions from this, leaving only 8.8% unaccounted. Okay, orthogonal linear combination of the output. That is what a principal component is. The point is to show as much variance as possible using few principal components. Of course, just like we did over here, we can do principal component 1 against principal component 2, principal component 1 against principal component 3, and 2 against 3. So if 2 dimensions doesn't give you enough of the total variance, you can add one principal component. But the point is still the same, right? If you have a 100-dimensional data set, and if you look at the first 3 dimensions, then with 3 2-dimensional plots, you can summarize 100-dimensional data. So it's a very useful tool. Okay, you can do other things with it, which you don't have to remember. Just know that that's a thing called principal component regression. And a lot of more complex ways to summarize high-dimensional data, a lot of multivariate techniques, they all rely on Pca. They start with Pca, or they do some generalized version of Pca, like multidimensional scaling. So Pca is really the foundation of many more advanced exploratory data analysis techniques. Okay, that brings us to the choices in Pca. There's really only 3 choices. There's whether you center, whether you scale, and how many principal components are you going to look at. So centering, it means that you remove the mean from each variable so that they are centered around 0. Scaling means that you divide each variable by its standard deviation, and that means that it will have a standard deviation of 1. So why would you do that? Well, centering, for the purpose of this course, is always done. There are fringe cases where you don't want to center, but in almost all cases you do want to center because the result is more difficult to interpret when you don't. So just remember that you should always center your data for Pca. There is an option to turn it off, but unless you know what you're doing, leave it on. Scaling is a different story. Whether you scale or not has a really large effect on what your principal component analysis will look like, and also how you can interpret it. If I divide everything by its variance, then it's all on the same scale. So that's why we call it scaling. But let's say that I measure all of my variables in millimeters, right? They're all physical dimensions or something. Then scaling will attribute equal importance to all of them, even though one may have much larger differences than the other. So then it's unwanted. However, let's say that one of my data set is the degrees in degrees Celsius. Another one is the length of a road in kilometers. The other one is the elevation in meters. The elevation in meters is going to have much larger numbers than the temperature in degrees Celsius. So these are on a very different scale. And remember, variance is just the squared difference from the mean. And if you square large numbers, then you're going to end up with the larger numbers. So the variance depends on what scale you measured something. If I measure something in meters, the variance is different than if I measure something in kilometers. So if this is the case, you have variables of different sources measured on different scales. That's what it's for, and then you should scale. If all of your variables are measured in the same way with the same unit of measurement, then usually you don't want to scale, because if you do, then you are saying that small differences are as important as large differences, which is distorting what's actually happening. There are some concrete examples of like problems where I would and wouldn't scale in the book, which I'll show at the end. Okay, last question is how many principal components should you use? Because of course you can just make a 2D plot of principal component 1 and 2, but yeah, what if that only explains 10% of the variance? Then how much further should you go, right? So there's two ways to do this. One is to think for yourself, how much of the total variance do I want to have accounted for, right? You could say I want 95% of the variance accounted for, or 90%, or 80%, whatever you're happy with. Another one is called the elbow rule. Now, the elbow rule is by producing a scree plot. It's not important that you remember it's called a scree plot, but it's useful to know because then you could know what the function is called, which plots the proportion of variance explained by every principal component. And then you can see, okay, if I only use one dimension for this example PCA, that gives me 46.5% of the total variance. If I also include the second dimension, then I have another 22.1%. Now, what does the elbow rule say? It says if at some point there's a sudden drop-off in how much variance it adds, that's where you stop. So, yeah, the problem is, of course, that's a bit subjective. You could say that the biggest drop-off is here. You would be right. But then I would say, well, a one-dimensional plot isn't easier to understand than a two-dimensional plot, so just go with the first two at least. But, you know, why add a third one? Well, okay, that is because personally I think that 46.5 plus 22.1 is still not so much of the variance. It's like, what is that, 70% or so? So that leaves about 1/3 of the variance unaccounted for. So I don't find that satisfactory. But then, this is a small step, right? This gets me another 12.7%. This still gets me another 10%. But then suddenly, this gives me less than 1/20th of the remaining variance. And after that, even less, and at the end, it doesn't add any information anymore. So, yeah, it's a heuristic. It's not a hard rule, but you can use this if there is a clear elbow, so, you know, shaped like this, where you stop adding extra dimensions to look at. And then you can plot them. You can plot PC1 against 2, 1 against 3, 1 against 4, etc. And of course, that's a lot of work, but it's a lot less work than looking at 100 different variables one by one. Okay, yeah, that was all I have to say about PCA. Now I'm going to show you how you can do it in R. This is an example from the book, so I will quickly download the book. It's over here in the link. I think there's also a link to it on Brightspace, otherwise I'll post it later. So we're just going to download this one. Yeah, and then in the book 4.4, so chapter 4 is about principal component analysis, and then 4.4 is examples of PCA in R. Now, these are two examples. The first one is just do PCA, I don't care about anything else. The second one is, okay, that looks really ugly, let's make it a little bit less ugly. So, how to use it, just copy it. This is the iris dataset, so it is 5-dimensional, but yeah, we're not going to use species because it's not numeric, so I cannot calculate a variance. So I'm only going to use the first 4 dimensions, the sepal width, sepal length, petal width, and petal length, and see what they look like. So I just copy this example and I paste it. If you want to know how it works, there's comments behind every piece of code of what it does, and then just run that. And that gives you a biplot. And then we can see whether this makes sense. Now, take some time to look at this. Okay, maybe not this, because this is the ugly one. Yeah, that's better. Okay, take some time. Well, not much actually. Okay, the legend looks really weird on my laptop, so I'll just turn off the legend. You'll just have to take my word for it. These are 3 different species, red, blue, and green. And these are the different variables, right? Yeah, so let's look at the ugly one anyway, because that one has the names over here. This says sepal length. This says sepal, is it S? Yeah, sepal width. This says petal width, and this says petal length. So what does this plot tell us? Anyone have an idea? Yeah? Yes, sepal width accounts for the largest part of variance. Yes, definitely. That's one of the things you can conclude. Why? Because it has the largest contribution to PC2 and PC3 in this case. We can also look at this one, which is PC1 versus PC. Let's go to PC1, PC2. Sorry, because that is the largest variance. But you are correct. The largest arrow, in this case pedal length, shows you the largest variance among all the variables. Okay, what else do we see? In case it isn't readable, this says pedal length, this says pedal width, this says sepal length, and this says sepal width. Anything else you notice? Yeah?
Can you say there's a correlation between petal length and petal width because it's going with the two vectors of petal length?
Exactly. So remember that we're finding linear combinations, and it can be the case that there's correlation among your variables. Now, if pedal width and pedal length, they point in exactly the same direction, then apparently they don't add unique information. And that's you can see that because the arrows are in exactly the same direction, right? So it means if my pedal width goes up, my pedal length goes up. In other words, we expect them to be correlated. Well, that's something we can calculate. So let me zoom in a little. Correlation of iris, and we're not going to look at species because that's a new factor, so that doesn't work. Okay, well, where do we see whether she was right or not? Is it readable? Too low, maybe? Oops. Still too small? I mean, I can make it as big as you want. Yeah, it's not much better this way, maybe like this. Yeah, there we are. So, on the rows we have the variables, on the columns we have the same variables. This is a correlation matrix, right? It shows how each variable is correlated. How can we see that there's high correlation or redundancy? Yeah? Yeah, careful with that one. Sepal length correlates perfectly with what? With sepal length, right? It's the same variable. But yeah, that's right. That one has a perfect correlation. Yeah?
It's closer to one, like the pedal length and pedal width.
Exactly. Yeah. So remember, the one that was perfectly overlapping is pedal width and pedal length. And that's exactly what we see here. We see that let's start here. Pedal width correlates with pedal length, that's row 3, with 96%. So they don't add much unique information. If your pedal length is longer, your pedal width is longer. That's one of the nice things about PCA. Because, again, imagine this was a 100 by 100 matrix. How are you going to look at all of those correlations and say something meaningful about it? Well, with PCA, you can just see there's a bunch of arrows in two-dimensional plot. Now, another thing, let's remove the arrows for a moment, or at least the names. Oh, yeah, we're figure margins. If you get this error, by the way, it just means that the plot window is too small. So, this one. What else can we see? I've colored by species. Where are you? Here. I don't know if the colors are visible. Are they? Okay. What's the first thing you notice? Yeah? Yeah, you can separate these species in this two-dimensional plot. Now, if you remember from some of the exercises, actually these two groups, if you look at one variable at a time, they have quite some overlap. But if you use all variables at once and then summarize the largest variance that there is in 2 dimensions, actually there's almost no overlap between these species. So you could use this for clustering. Yeah, you could see, okay, are my species at all separable using this set of variables? Yeah?
The Earth still makes sense if you see different groups.
What do you mean? Why would the arrows not make sense? Okay, I can show you how to interpret this. Yeah, so this was sepal. One of them is petal width, one of them is petal length, right? The two correlated ones. What does this mean? This means that the red group, that one is the furthest in this direction, has the lowest petal width and petal length. And this group as is in the middle, and this is the highest. The green group has the highest petal length and petal width, right? And the other thing we can see is that species are more separated by their petal dimensions than by their sepal dimensions. Yeah, let me go back to the one. Sorry, I should have added notation to the plot, but we can work with it. Yeah, just imagine this is red. You can see that the separation of species is more determined by the petal dimensions than by the sepal dimensions, right? Because sepal dimensions are down, petal dimensions are to the side, and along this x-axis there's much clearer separation of them than along the y-axis.
But could you not expect that petal width to maybe be different among the group, between the group?
Yeah, it is.
But
What do you mean?
well, now you basically have like the petal width is basically the same as the petal length.
Yeah.
If you see that you have different groups, couldn't that correlation be different unless like your interpretation could be wrong? Wouldn't you say?
Okay, so this shows the contribution of these variables to this coordinate system. So of course they are wrong. The question is by how much. Now, how much variance does this PCA explain? Let's see. Yeah, I wrote it over here. This first dimension shows us 92.5% of the variance. So yeah, okay. Oh, sorry, this is the ugly one. Yeah, this is 92.5% of the variance. So yeah, is it wrong? Yeah, but only by 7.5%. All of the other variance is accounted for. So the difference in petal dimensions is, yeah, for 92.5% captured by this direction. Yeah? So what do we use it for? Summarizing high dimensional data. You can use it for other things, but you don't have to know that. Okay, I'll show one more time where to find this. It's over here. Page 65. You can click on the links if you download it. And yeah, just copy the code. If you want to know what an individual piece of code does, there's comments written besides it. Take note that it produces several plots. Yeah, so this is PC1 versus 2. This is PC1 versus 3, 2 versus 3. And also we get a scree plot. So anyone remember what this one says? This is the default one, so it doesn't come with numbers. But what does it say? Yeah? No hand? Yeah, this is the one with the elbow rule. So what is this first thing over here? What does this mean? Yes, and what does the size mean? Yeah, this is the proportion of variance. Okay, here it's not a proportion, but the actual number. But if we compare them to the rest, we can see how much variance did PC1 explain, how much did 2 add to that, how much did 3 add to that, and 4. And again, we can see the same thing we saw when I plotted it. This is already 92.5%. You can visually kind of see it's about 90%, right? And this is much less, and this is almost nothing. Yeah, so it means you can safely discard 2 dimensions, and you'll still have like 95% of your data accounted for. Yeah?
For example, the two are correlated and also there's a big variance. Does that mean that like if you add a second PCA it doesn't explain more in the percentage wise?
Yeah, you're almost there. If you have correlated variables, that means there's redundancy, right? Because the information explained by one of your variables is also in another. The petal width and the petal length, they have such a strong relationship that if I know one, I know the other. That means that the first principal component will capture the relationship from either one of them. Second one doesn't add anything. Okay, what then happens? It means the last principal component will be 0%, right? Because we also see it over here. This is like, well, I can call it in numbers. Let's see the code for a moment. Yeah, variance explained. Okay, let's see how much variance is explained by each principal component. The first one explains 92.5%. The last one only 0.5%. Yeah, so that one adds almost nothing in variance of your data. Why? Because there were two very strongly correlated variables. So you really don't need 4 dimensions to show the variance in this data. Yeah?
Do you need at least 2 PCs or is just one good enough?
You could use one, but the thing is, if you're gonna make a, oh, let me see, where is it? Oh yeah, sorry, here. Yeah, let me show you what a one-dimensional plot looks like. Here, like this. What does it hurt to add a second dimension? You know, if the purpose is to make a plot, just use 2 dimensions, you know? But yeah, if you want to do some form of regression with it, which you can do, then of course you can use only one if that's already enough of the total variance. Yeah, let me check if there's other questions online. Oh, yeah, someone asks, okay, if there's still people without access to Brightspace and you want to have the solutions, please send an email because I don't know who still doesn't have access, and then I will send you the solutions. Okay, any other questions about this topic? If not, then let's check the schedule. Yeah, we have until 1:15. You know, of course, you take a break in between to work on the assignment. Just see if you can find any interesting things going on, like is there correlation? Can I somehow cluster my data? Just try plotting it. Yeah, there's 3 data sets. You can try applying it to older data sets, and you can see how informative or not informative it is. Yeah?